Hello, my name is Hong Mu. I'm a postdoc from Professor Susie Broy's lab in the biology department of New York University. Here on behalf of our collaborators, Professor Yingkai Jiang and Professor Nicholas E. Gicinto from the chemistry department of New York University and Professor Jung-Yoon Min from the chemistry department of University of Illinois at Chicago, I will present our work entitled The Nucleotide Excision Repair Lesion-Recognition Protein, RAD4, captures a pre-flipped partner base in a benzo A pyrene-derived DNA lesion. How structure impacts the binding pathway. Nucleotide excision repair (NER) uses a cut-and-patch mechanism to excise many DNA lesions. The NER machinery excises a 24 to 32 nucleotide long oligonucleotide that contains the lesion. The gap DNA sequence is restored by DNA polymerases using the undamaged strand as template. NER deficiency causes human diseases, including xeroderma pigmentosum, which leads to extreme sun sensitivity and skin cancer. Hence, it's very important to understand the NER mechanism. Furthermore, understanding the mechanism can help us to design more effective chemotherapeutic drugs, since the potency of these drugs can be impaired by NER. XPC is key in initiating lesion repair by NER. It recognizes a wide variety of DNA lesions. In order to initiate successful subsequent NER, XPC needs to bind productively to lesion-containing DNA. The structure details of the productively bound complex have been revealed by the crystal structure of RAD4 in complex with a CPD-containing DNA, solved by Min and Pavletich. RAD4 is the yeast ortholog of human XPC. Shown on the bottom left are the chemical and 3D structures of the CPD lesion. The CPD lesion can be induced by UV light in the sunlight. It can cause skin cancer. Shown on the right is the structure of RAD4 productively bound to a CPD-containing DNA. There are 4 domains of RAD4 shown in the figure: the transglutaminase domain, TGD, and 3 beta-hairpin domains, BHD. TGD and BHD1 are shown in yellow. They bind non-specifically to the DNA sequence away from the lesion site. BHD2 is shown in orange, and BHD3 is shown in green. Most interestingly, BHD3 has a beta hairpin inserted into the helix at the lesion site, separating the CPD lesion from its partner strand. And BHD2 binds on the minor groove side of the lesion site without intrusion. The CPD lesion, which is shown in red, is extruded into the major groove, and 2 bases opposite the lesion are flipped into the protein binding pocket. It's very interesting that NER efficiency varies greatly among different lesions, influencing their toxicity and mutagenicity in cells. Some lesions are even repair resistant. These can survive to replication and cause mutations that initiate cancer. However, the origin of such great variability in NER efficiency is not understood. Our study focuses on the mechanism of a crucial step in NER, lesion recognition by XPC. We hypothesize that different lesions whose impacts on DNA structure and stability vary manifest differences in binding pathways and their energetics. In this work, we investigated binding pathway of RAD4, the yeast ortholog of human XPC, to a well-repaired bulky cis-BAP-DG adduct. This covalent DNA adduct is derived from the environmental pro-carcinogen benzo A pyrene, which is present in automobile exhaust, tobacco smoke, among others. Shown on the bottom left is the chemical structure of the cis-BAP-DG adduct. The enema structure of a cis-BAP-DG containing DNA duplex with a normal partner base C was solved in a collaborative effort with the Patel Lab in the Memorial Sloan Kettering Cancer Center. It adopts a base-displaced intercalated conformation. Shown in the middle of the bottom is the central fiber of this NMR structure. The BAP rings are shown in red, the damaged guanine and its normal partner C are in blue. The BAP rings are intercalated in the DNA helix, where the damaged guanine is displaced into the minor groove, and its partner C is pre-flipped into the major groove. Compared with our previous study of the small CP deletion, our results show that the pathways differ significantly for these structurally different lesions. We obtain the preferred molecular binding pathway and its associated free energy using molecular dynamics simulations with umbrella sampling for free energy calculation. Our initial models were based on the NMR structure of cis-BAPDG lesion and the crystal structures of RED4. The first capture state is achieved from unrestrained MD of initial model. Starting from the first capture state, the structure of the complex was guided to achieve the productive binding state, where the BHD3 beta hairpin is inserted and the partner strand bases are flipped into the portin binding pockets. We explored many binding pathways using umbrella sampling with restrained MD simulations. Free energy calculations were performed using VFAP method. The pathway with the lowest free energy barrier is chosen as the preferred molecular binding pathway. Our MD simulations were carried out using AMBER 14 package with FF14SB force field for portin and DNA. We applied customized force field for the cis-BAPDG lesion. The complete free energy profile for the binding pathway of RED4 to the DNA duplex containing the cis-BAPDG lesion is shown on the upper right. A structural movie is provided in supporting information. The snapshots of key states along the pathway are shown at the bottom. These structures are viewed from the minor groove side of the lesion site. At the first capture state, BHD2 hairpin binds into the minor groove near the lesion site, and the pre-flipped partner C is captured by the portin, where it stacks on top of phenylalanine 599 of BHD3. Starting from the first capture state, BHD3 hairpin approaches from the major groove. It further distorts the lesion site to bend and unwind around it. Such distortions start to rupture a base pair flanking the lesion site. At the transition state, a second base has flipped into the portin following the rupture of its base pair, and the BAP rings began to extrude. From the transition state, BHD3 beta hairpin begins to insert, while BAP extrudes further. The two flipped partner strand bases move along the flipping path that utilizes 3 phenylalanines to guide the bases into their binding pockets. The 3 phenylalanines are phenylalanine 599, phenylalanine 597, and phenylalanine 556. At the intermediate state, 2 partner strand bases have moved to phenylalanine 597, and they are ready to be fully inserted into their binding pockets. Starting from the intermediate state, BHD3 beta hairpin further inserts to fully extrude the BAP rings, while the partner bases insert completely into the binding pockets to achieve productive binding state. At the productive binding state, phenylalanine 599 stacks with a base pair flanking the opened up lesion site, where the lesion is extruded into the minor groove, sandwiched in between the DNA backbone and the beta hairpin tip of BHD2. The 2 partner strand bases are fully inserted into their binding pockets. Structural differences of CPD and cis-BAPDG containing duplexes explain their different binding pathways. Shown on the left is an overlay of the damaged duplexes in their initial models. The CPD containing duplex is shown in light gray. The thymine dimer part of the CPD is in purple. The cis-BAPDG containing duplex is shown in dark gray, where the BAP rings are in red. The damaged guanine and its partner base C are in blue. In the cis-BAPDG case, greatly enlarged minor groove and pre-flipped partner strand C promotes initial capture of this base. In contrast, CPD does not have a pre-flipped partner base and has a narrower minor groove. The initial binding involves only the probing by the BHD2 hairpin tip in the minor groove. For the cis-BAP-DG case, intercalated bulky BAP rings require tightly coupled BHD3 hairpin insertion and BAP extrusion for productive binding. However, for the CPD case, the small lesion extrudes readily to the major groove, while the partner bases flip in correlated motion into the porting, leaving a fully opened site. Then the BHD3 hairpin inserts spontaneously. In conclusion, we have determined how the base displays the intercalated cis-BAP-DG supports a binding pathway that differs from that for the small CPD lesion. For the cis-BAP-DG, conformational capture of the pre-flipped partner C plays an important role in the initial binding to the damaged site. Our work also highlights that XPC may achieve versatility in productively recognizing different lesions for NER by employing different binding pathways. Our work was supported by National Institutes of Health and National Science Foundation grants. Our computational resources were supported by extreme science and engineering discovery environment and high-performance computing resources of New York University.